Hello everyone, welcome to today's class. Today we are going to discuss principles of biotechnology. So if we see the principles of biotechnology, the two principles of biotechnology are genetic engineering and bioprocess engineering. Now let's see genetic engineering. It is basically a technique that alter the chemistry of genetic material. Any genetic material which is naturally provided, if we take out that genetic material and make some changes, either by addition of foreign materials, we either by addition of foreign materials or by introduction or by introduction of genes from one organism into another, we call it genetic engineering. So basically it's a technique that alter the chemistry of genetic material. So if this is the DNA, now this DNA we have added into a bacterial cell. Now this DNA will combine with this plasmid, extra chromosomal segment of DNA, and now it is called as a recombinant DNA. So here the DNA is being modified. So this genetic engineering basically deals with the genetic material. Modification in the genetic material we call the genetic engineering. Coming to bioprocess engineering, it is basically maintenance of sterile environment so that only desired microbes grow. For example, if we want to isolate, if we want to produce vaccines, or if we are doing, if we want to grow certain microbes, if we want to grow a very specific microbes, we need to maintain the environment sterilized so that other microbes do not grow along with our desired microbes. So this process of maintaining a sterile environment so that only desired microbes or desired process takes place smoothly, we call that bioprocess engineering. For example, antibiotic production, vaccines. So when we are working with antibiotic production, we need specific microbes to grow, right? So and we, for that reason, we need to maintain the environment sterile so that other unwanted microbes do not grow. So that process of keeping the environment sterile for growing only a desired microbes, we call it bioprocess engineering. Coming to gene cloning. What is gene cloning? Gene cloning is making multiple copies of the gene. Gene cloning is a natural process of making multiple copies of the gene. Same making multiple copies of the gene if we do it in lab. In lab, it will be called as PCR. Making multiple copies of gene, naturally we call it gene cloning, right? And if we do it in the lab, we call it PCR. So what are the steps? We first isolate a desired gene. If this is a bacteria and this is plasmid, right? And say this is any gene we have taken, any DNA containing desired gene, DNA containing desired gene. So here isolation of desired gene. So first this is present in a cell, right? This is present in a cell. Now we need to isolate this DNA. Isolation of DNA, right? Construction of recombinant vector. Now we will take plasmid. That is what we call, this is a plasmid which is acting as a vector. Now both of them combine and leads to the formation of a new recombinant DNA. So isolation of desired gene. So we have isolated this DNA from a cell, right? Then construction of recombinant vector. This is what is a recombinant vector, right? Then we put this back into the bacterial cell. Now once you put it back, once you put this recombinant vector back into the bacterial cell, whenever the bacterial cell grows, this vector also gets multiplied. So here what will happen? Multiplications of recombinant DNA, right? Leading to host increase or leading to the increased production of gene, right? So this is what we call gene cloning. Now if we discuss about the first recombinant DNA, so first recombinant DNA was done, the first recombinant DNA was made by Stanley Cohen and Herbert Boyer. These two scientists, what they took a bacteria known as Salmonella typhi, which causes typhoid. So they have extracted the plasmid. This is an extra chromosomal segment of DNA. Plasmid is what? It is extra chromosomal segment of DNA. So this plasmid, they have extracted the plasmid from the Salmonella typhi, and in that plasmid they introduced a new gene. The name of the gene is antibiotic resistant gene. So they have inserted this antibiotic resistant gene into the plasmid, and this is known as recombinant plasmid. And this they cannot put back into Salmonella typhi because otherwise it will show resistant to the drug. So we cannot, otherwise if we include this, if we insert this recombinant plasmid to Salmonella typhi, then what will happen? This will be showing resistant to that particular antibiotic, hence treatment of typhoid will be difficult. So what they did, they inserted this plasmid into another bacteria, we call it E. coli. So this is the case how the first recombinant DNA is being produced. Coming to restriction enzymes. Restriction enzymes are enzymes which are needed for genetic engineering. Main enzyme needed for gene modification. This is the main enzyme needed for gene modification, right? So this is also known as molecular scissor. This is used to cut DNA. This is also known as molecular scissor because this enzyme is used to cut DNA. It is obtained from prokaryotes. 900 restriction endonucleases have been isolated from 230 strains of bacteria. The first restriction endonuclease that has been produced is HIN2, and it recognizes 6 base pairs. So let's discuss restriction modification system. It's a concept in which if this is a bacteria, So when a virus infects a bacterium, what bacterium will do as a defense mechanism? Bacteria will produce an enzyme known as restriction endonuclease. What is the function of this enzyme? This enzyme cuts the genetic material, DNA of bacteriophage, by recognizing certain specific sequence. Now, this specific sequence will also be present in the bacterial chromosome. So, how bacteria will protect its own DNA? To protect its own DNA, what bacteria will do? Bacteria will produce another enzyme known as methylase. What it will do? It will add methyl group to the bacterial recognition site. If suppose CCC is the site which a restriction endonuclease recognizes and cuts, then this CC site here in virus, the restriction endonuclease will act and cut. But in case of bacterial cells, methylase, an enzyme will come, it will add a methyl group to this recognition site, thus protecting its own DNA from restriction endonuclease. So this concept we call it restriction modification system. Why we call it restriction modification? Because the first enzyme that has been produced is restriction endonuclease as a defense mechanism. So we call it restriction modification because here bacteria modify its own DNA by adding a methyl group with the help of methylase enzyme. So this concept is known as restriction modification system. So with now we will see the naming of restriction enzymes. So restriction enzymes are always named with echo R1. E stands for genus, O stands for species, R stands for strain, and this is a number, Roman numbered, and this has been according to the order in which enzymes are isolated. So the first letter is basically stands for genus, second stands for species, and R, here R stands for the strains, and number is according to the order in which enzymes are isolated. With this, we would like to end this today's class. Next class, we will see tools of recombinant DNA technology. Thank you so much for joining this session.